Hello everyone and welcome to BMLT Education Point. Today we are going to dive into one of the most revolutionary techniques in modern molecular biology, PCR or polymerase chain reaction. Think of PCR as a molecular photocopier. It allows us to take a tiny, almost invisible fragment of DNA and amplify it into millions of copies so we can study it in detail. The three essential steps. To understand how PCR works, we need to look at its three-step thermal cycle. This process happens inside a machine called a thermal cycler. 1. Denaturation, the unzipping. First, we raise the temperature to about 95 degrees Celsius. At this high heat, the double-stranded DNA template melts or separates into two single strands. This exposes the genetic code so it can be copied. 2. Annealing, the priming. Next, we cool things down to between 50 degrees Celsius and 65 degrees Celsius. This is the annealing phase. Here, small pieces of DNA called primers bind to specific target sequences on the single strands. These primers act as markers, telling the enzyme exactly where to start binding. 3. Extension, the building. Finally, we heat it back up to roughly 72 degrees Celsius. This is the optimal temperature for Taq polymerase, our star enzyme. It grabs free-floating nucleotides, DNTPs, and builds a new complementary strand of DNA. By the end of this step, we have two identical double-stranded DNA molecules. Key components and procedure. But what do we need to make this happen? As you can see on the poster, our master mix includes the DNA template, the sample we want to copy, primers to start the reaction, DNTPs, the building blocks, and Taq polymerase along with a specialized buffer to keep the environment stable. The general laboratory workflow starts with DNA extraction.